Hello and welcome to PH Biology. Today's explainer is the prokaryotic cell. No nucleus. DNA floating loose in the cytoplasm. Tiny. By the end of this video, you'll know what makes a cell prokaryotic, why plasmids matter for biotechnology, and exactly how prokaryotes differ from eukaryotes on the GCSE paper. A prokaryotic cell has no nucleus. Its DNA sits free in the cytoplasm, often joined by small extra circles called plasmids. There are no membrane-bound organelles like mitochondria or chloroplasts. Prokaryotic cells are tiny, typically 1 to 10 micrometers, 10 to 100 times smaller than eukaryotes. Bacteria are the textbook example of a prokaryotic cell. Three things to remember. 1. There's no nuclear membrane, so the DNA is naked in the cytoplasm. 2. Generally no membrane-bound organelles. And 3. They're tiny. They do have cell walls, ribosomes, sometimes flagella for movement, and plasmids that can be transferred between bacteria, which is exactly why genetic engineering uses them. Common mistake. Prokaryotes definitely have DNA, they just don't enclose it in a nucleus. And don't confuse prokaryotic with simple. These cells have complex chemistry, including the ability to mass produce human insulin when we engineer their plasmids in the lab. Quick example, E. coli is a typical prokaryote. A single cell, no nucleus, plasmid DNA, surrounded by a cell wall. We use E. coli extensively in biotechnology. Insert a human gene into a plasmid, put it back into the bacterium, and the bacterium becomes a tiny factory producing human protein. Right, if any of this is going to stick beyond this video, drop into phmaths.com. You'll find a free 4-week GCSE higher revision planner ready to download, plus the mastery workbooks, one for every higher topic, every answer fully worked, every common slip flagged. Simple explanations, serious results. See you next video.